back to humans again. So our next presenter is Tingshan Liu, who just completed her master's at the University of Chicago, which is also my alma mater. So I'm very pleased to see you here, and I miss Chicago. So welcome to this panel. And Tingshan is going to present her exciting work on amygdala connectivity across the schizo-bipolar disorder, specifically looking at facial recognition. Thank you very much.
Hello, everyone. So I'm very happy here to present the project that I was working on while I was still in school. So in this project, in this presentation, I will talk about the amygdala functional connectivity across the schizo-bipolar spectrum and its relationship to facial emotion recognition. For people who are not very familiar with the term schizo-bipolar spectrum, it usually incorporates patients with DSM diagnostics of schizophrenia, schizoaffective disorder, and bipolar disorder with a history of psychosis. So some of the background information. So a lot of people can have observed people with a history of psychosis have a diminished social functioning, and a lot of people think it might be related to their facial emotion recognition deficits. Indeed, there is actually large literature has reported facial emotion recognition deficits in schizophrenia, and is now considered a well-established track marker for schizophrenia. There are a little bit less work on schizoaffective and bipolar, but in general, they also suggest that schizoaffective patients also show some sort of impairment, but less than schizophrenia. There are some mixed findings for psychotic bipolar group. It usually varies depending on the illness state or their mood state of the patients that they conducted analysis on. So the question about what is the neural basis for recognizing facial emotions, this is very very big question, and and also there are multiple areas brain areas are involved in this very complicated process, from perception to final recognition of the emotion. The process might involve brain structures such as amygdala, for fusiform face area. So that attempts to localize the perception or recognition of the stimulus in space or in time really suffer from the fact that the same brain structures participate in different components of processing at different points in time. So in this project, we can try to understand this process using neuroimaging methods, more specifically resting state fMRI data. From previous studies, a lot of studies are pointing towards the amygdala connectivity dysfunction in patients with schizophrenia, both from resting state as well as look at their activity in tasks. So that's the main reason that we also decide to look at the amygdala functional connectivity in this project. The main research question for us would be, first, do patients with psychosis exhibit altered resting state amygdala functional connectivity comparing to healthy controls? And if so, is this alteration associated with their facial emotion recognition deficits? So to answer this question, here are some of the material and methods that we used. So all samples are drawn from the BSNIP2 study. This is a very large, multi-site study that is trying to find biomarkers to better classify the phenotypes across the schizo-bipolar spectrum disorders. So all of the samples that we have completed a behavior task, which is a dynamic affect recognition evaluation task. and they also completed a resting state fMRI scan. So I'll briefly go over this task. This is, we call it a dynamic facial emotion recognition task, because basically the patients or subjects, they will watch a very short video where the faces transition from a neutral to like one of the 6 emotion categories, including happy, sad, anger, surprise, disgust, and fear. So as soon as they know what the emotion type is, they will press the button and then choose the answer from one of the 6 categories, basically. So therefore, so this task will allow us to have 2 variables of interest. One is emotion recognition accuracy, and the other one is latency. So basically, accuracy and speed both matters. So some of the like very basic demographics for the sample that we included for the final analysis. So the final sample size is 346 patients, like with over 100 healthy control, and also over 100 schizophrenia patients, 84 schizoaffective, and like a smaller sample size of psychotic bipolar patients. This is the final sample size after we screen out, like after the pre-processing, we screen out the bad scans or like patients with too much movements. And so these are like with good quality of data, and to put it that way. So I present age and gender here because these are the 2 main covariates that we included during the fMRI analysis, which I will present later. But before that, I will also show the very basic information about the behavioral data. So here I present the total emotion recognition accuracy score and their total latency score, like how fast they identify facial emotions. So on the left of the panel is the total accuracy score by each diagnostic group and healthy control. Schizophrenia has a significant lower accuracy score than psychotic bipolar group and healthy control. This is largely like consistent with a lot of the other previous studies where schizophrenia has the largest deficits in terms of recognizing the emotion accurately. On the right of the panel is the total latency score. Both schizophrenia and schizoaffective are significantly lower than healthy control in recognizing emotional phases. So here I didn't, well, in the real analysis, we definitely break it down into to look at the group differences in terms of like recognizing each type of the emotion phases. However, we only include the total accuracy score and the latency score for the mirror imaging analysis. A main reason arguing for that decision would be that so far there is no literature or study suggesting that like the brain, like different brain areas are recognizing different like discrete type of emotions. So we care more about the total score less than about the scores for each type of emotion type paradigm. So here is some of the results from the C-based functional connectivity analysis. And we looked at left amygdala and right amygdala connectivity individually. So the first step is to do a F test. We would like to see if there are any significant differences on amygdala connectivity between any diagnostic groups. And after controlling for age, sex, FD power, which is a motion indicator, and scanner sites, there is no significant differences across these 3 diagnostic groups. Therefore, we decide to combine them together and then compare if there are any differences between patients overall versus control. And comparing to controls, we found that patients actually exhibited decreased connectivity in bilateral insular cortex, right planum temporal, and right supramarginal gyrus to the left amygdala. And there is also decreased connectivity in right planum temporal and right insular cortex to right amygdala. All the significance are family error wise adjusted. So now we see that there are some amygdala connectivity deficits in patient group. We want to try to answer the second question of the project is whether such altered amygdala functional connectivity is associated with any part of the behavior task. So we tried, so there is no significant correlation between accuracy score or latency scores and amygdala connectivity across all subjects. And this is still true after discounting the differences in average connectivity between patients and controls. And we also look at this correlation within each diagnosed group and healthy control, but we do not find any significant correlation between amygdala connectivity and accuracy. We also did not find significant correlation between amygdala connectivity and latency score in schizophrenia and bipolar group. However, in schizoaffective disorder group, there is actually a significant positive correlation between latency score and right amygdala connectivity to right middle frontal gyrus. The significance is very large, like even after like accounting for multiple comparisons. So just some recap of this whole project. There is a reduced amygdala functional connectivity observed in patients group, and this might serve as a transdiagnostic rather than illness-specific biomarker for schizo-bipolar spectrum disorders. However, we still need more bipolar sample in future studies in order to validate this, conclusion. And facial emotion recognition accuracy did not correlate with amygdala functional connectivity, both across the groups and within each group. And in patients with schizoaffective disorder, there's a significant positive correlation between latency and amygdala functional connectivity to middle frontal gyrus. So I think for the, we still kind of like want to do more research to not look at resting state functional connectivity, but also functional connectivity while the patients are doing this emotion recognition task. Those are also like a very helpful to understand the neuromechanisms of this facial emotion recognition process in patients. Okay, that's my.
Wonderful. Thank you so much for a fascinating talk. We only have one official minute for questions, and then we get kicked out in 5 minutes. So are there any questions from the audience? If not, I'll ask a question. You were talking about emotion accuracy. I don't know if I understood your task correctly. It was morphing from neutral to fear, neutral to angry, or was it morphing between emotions?
No, it's just one type of emotion for each trial. So neutral to happy for a trial, for example, and then maybe neutral to angry, and it's kind of like randomized emotion types.
Right. So it's detection, because accuracy, I guess, is subjective, maybe, right? Like,
yes,
all of them kind of contain an emotion. So I guess early detection.
Yeah.
or something like that. I wonder if you morph them between emotions. So like if you morph between happy and fearful, or happy and angry, if you would get something different, because there you're pitting emotions against each other.
Yeah. Yes.
Mm-hmm.
Yeah, I definitely agree, because I personally also have like some of the thoughts about the task itself, because like it always like basic emotions, but really there are a lot of like still a lot of arguments about that, and this is really more of a cognitive task, because there's a correct answer for emotion type, but there are a lot of them look very similar, like surprise versus fear and anger. Like a lot of people will make mistakes about those things, and subjective in some way.
Mm-hmm.
So yeah, which also yeah, we need more research about this, and also some more research about the task itself, I guess.
Well, it's great that you had it animated. I think that's an advancement over some of the still paradigms. Thank you very much for this talk. Are there, we will get kicked out in 3 minutes. Before that, are there other questions maybe from audience members? As a developmental neuroscientist, I would be really eager to see this kind of thing in maybe at-risk siblings or children. I think that would be really informative to see sort of the extended phenotype, or siblings or something like that at a younger age, to see some causality, or maybe see some of the precursors that lead to these connectivity differences.
Yeah, yeah, for sure. And also a lot of like risk for, say, like early onset psychosis, like children or like young adults, they exhibit like eccentric behavior sometimes, and also lead to like social, I don't like to say deficits, because it doesn't, yeah, sometimes I feel like there also something to do with the environment. It doesn't necessarily to do with the.
Yeah, absolutely. I agree. I like that perspective. I agree. Thank you for that. Well, thank you very much. You all had such complimentary, exciting talks.